Jeg synes, hvad I selv synes bedst.
Nej, det skal være udvalgt.
Okay. Skal vi tage på engelsk?
Okay.
Bienvenido.
Skal vi lukke lyset?
Ja, det tror jeg. Eller i hvert fald, ja. Jeg ved ikke, om der er flere muligheder end on-off. Det er der sikkert ikke. Vi kan prøve. Nej, det gør der. Jo. Okay, er du klar?
Ja.
Yeah.
Yes, I'm ready.
So let me start by welcoming you to Jens's master's thesis defense. Our opponent today is Professor Jeppe from Aarhus University, and the process, as I'm sure you know, will be that Jens will first give an overview of his work, and we'll probably take a few minutes break just to give everybody a chance to draw some fresh air. Might be necessary at that point. And then we will, it's question time, and Jeppe will start, and I will supplement, and we'll take it from there. And in an hour and a half or so, it'll all be over. So, okay.
Good afternoon, and welcome to my master's thesis defense. I'm Jens Morgensen, as you know, and the title of my project is the Net Acid Extreme Transporter NBCM1, identifying novel binding partners and characterizing phosphorylation sites of the SLC4A7 gene product NBCM1. The outline for my presentation today, I'll first start telling you about the introduction, a little bit about intracellular pH regulation, NBCM1 in general, then I'm going to the hypothesis names of this project, then I'll split up the results into two sections of this project, the identifying novel binding partners, and then phosphorylation site characterization. I'll round this all off with the conclusion, and then future perspectives. So, some theory. pH and intracellular pH regulation. First of all, pH is defined as the negative logarithmic of the concentration of protons inside a cell. A cell has an ideal intracellular pH of 7.2 to 7.4, but this can vary due to several changes, and they can either be regular or pathological. A regular change, for example, would be increase in lactic acid production during anaerobic respiration during exercise. Pathological would be it's a hallmark of, for example, cancer, where you have dysregulation of pH, an increased extracellular pH, which is lower, and alkalized intracellular pH. And due to these changes, cells have internal pH regulating mechanisms, two of which are buffering. The two systems are buffering systems and removal of acid from the intracellular environment. I must say the buffering system can deal with short and acute changes of pH, and here I include the most important one. I highlight the bicarbonate and carbon dioxide buffering system, which has the following equilibrium, which by the hydration of carbon dioxide with bicarbonic anhydrase leads to the creation of bicarbonate and H and protons. The other system is the net proton removal by membrane transporters, and there's a wide range of them, which I'm going to go through here in the next slide. Here we have a schematic of a cell membrane, we have the extracellular and the intracellular environment. Here we have the buffering system I talked to you about in equilibrium, with the CO2 being able to traverse the plasma membrane readily. The other charged species can't. For pH regulating transmembrane transporters, first we have NBCs, which are sodium bicarbonate cotransporters. By importing bicarbonate, they neutralize acid or extrude acid. Then we have anion exchangers, which exchange bicarbonate for chloride. By the action of extruding bicarbonate, they are acid loaders. And then we have NEGs, which are sodium hydrogen exchangers, which, as you can see, extrude acid. Then we have MCTs, which are monocarboxylate transporters, which export both H+ and lactate. Last but not least, we have V-type ATPase, which by the use of energy of ATP can pump hydrogen. Now I'm going to focus a little bit more on specific NBC, which is NBCM1. NBCM1 is an electroneutral cotransporter of sodium and bicarbonate, since it imports one ion of each. And since sodium is plus and bicarbonate is minus, there's no net change of charge. It's a member of the SLC4 gene family, which includes other anion exchangers and NBCs. We have identified several binding partners. There have been identified several binding partners, but yet the phosphorylation sites on NBCM1 are uncharacterized. A little bit more about MBCN1. When it's expressed in polarized epithelia, it usually has a basolateral ubication, and it's found expressed in a wide range of different tissues, like the brain, the heart, circulation, kidneys, digestive system, eyes and ears, amongst others. So it's widespread throughout the body in PHI regulation. Here we have there so far have been identified 16 splice variants. As we can see them over here, the first 16 are the ones that have been identified. The last two are truncated versions which have not been identified in vivo yet. And the 5 optional structural elements possible in MBCN1 are, it can either start with MEAD or MERF due to different promoters, and then it has also 4 splicing cassettes, 1 to 4. 1, 2, and 4 are located in the N-terminal domain, whilst 3 is located in the C-terminal domain. I'm going to explain that now with the structure, the hypothesized structure of MBCN1. And I want to point out that the longest variant of MBCN1 identified so far is MBCN1B, which includes cassettes 1, 2, and 3, and starts with MEAD. Here's the hypothesized structure of MBCN1. It has not been identified yet by crystal structure, but due to homology between the other members of its family, namely anion exchanger 1 and MBCE1, this is the structure. I'll go through quickly the parts of it. First, we have a long N-terminal cytoplasmic domain, which has 2 core subdomains and a loop region in between, followed by 10 to 14 transmembrane domains. I'd like to point out that it has a long extracellular loop between transmembrane domain 5 and 6. And then last, it has a shorter cytoplasmic C-terminal tail. Another structural element, hypothesized structural element, let's say expression element, is dimerization of MBCN1 and possible oligomerization of MBCN1 through interactions of its N-terminal linking domain. So why do we want to look at MBCN1? Why is it important? First of all, 2 single-nucleotide polymorphisms have linked MBCN1 to hypertension and cardiovascular disease in people of European descent, and also breast cancer in women of European and Asian descent. It's also known that knockout of MBCN1 in mice leads to combined deaf-blindness, which is known as Usher syndrome, and it's implicated in a role in this syndrome, the mutations at least. Now I'm going to go through the hypothesis names of this project. Hypothesize that MBCN1 as a memory transporter is regulated by interacting partners and also site-specific phosphorylations. To test this hypothesis, I used the rat MBCN1D, which is, remember, was the longest variant identified of MBCN1 so far. And I also want to point out that it has a 96% amino acid identity similarity to human MBCN1. And the 2 overall aims of my project, they were to, first of all, elucidate novel N-terminal binding partners, and second, to characterize MBCN1 phosphorylation sites. Part 1. So, I want to go through quickly what we know of MBCN1 and MBCN1 binding partners. Here we have the structure that I showed you before. For the N-terminal domain, it's hypothesized that MBCN1 interacts with IRBIT and calcineurin. Calcineurin more specifically binds only and interacts only when cassette 2 is expressed, MBCN1. And then for the C-terminal tail, there's 3 binding partners identified, namely hormonin, postsynaptic density protein 95, and EBP50. These interactions all occur around the PD set motif, which is the 4 last amino acids of MBCN1's amino acid sequence.